This video demonstrates the cold plating technique used in the backtalk cell signaling kit. Cold plating cultivates two bacterial species on the same plate to study how they affect each other's growth and behavior. The goal is to create two lawns of bacteria separated by a 0.5 centimeter zone of sterile agar. First, look and become familiar with the orientation of your agar plate. Note on which side the agar is poured and which side has the lid. It is important to use the alignment template provided to ensure there is a 0.5 centimeter gap between the different strains. With the agar side up, place the plate over the template. Using a permanent marker, label your plates with the appropriate information and the areas where each strain will be streaked. Using a straight edge, trace over the alignment lines provided by the template to mark the location of the 0.5 centimeter gap. Alternately, you can place the plate agar side down on the template while you streak. Gently flick the bacterial cultures to resuspend the bacteria prior to use. Use a micropipette and a clean tip to add 20 microliters of the specified strain in the center of the corresponding area on the plate. Dispose of the tip in the biohazard waste receptacle. Use a fresh inoculation loop to spread the bacteria within its designated area. Gently glide the loop across the surface as though painting the surface. The goal is to evenly spread the bacteria, but not go past the dotted line. Dispose of the loop in the biohazard waste receptacle. When finished, rotate the plate so you can continue to streak with your dominant hand. Repeat this process with the second bacterial strain. Allow your plates to dry for approximately 15 minutes until no liquid is observed. Then stack the plates agar side up and incubate as instructed. After 24 hours of incubation, your plates will be ready to view and analyze results.